At the cellular level, Alzheimer's disease is a mechanical failure. Synapses disconnect, and dense neural networks slowly fray until they physically collapse. Before we analyze lifestyle interventions, diet, or vascular health, we have to isolate the foundation of the disease itself. We begin with the non-modifiable variables, the immutable demographic and genetic factors that predetermine a patient's neurodegenerative trajectory. Understanding this unchangeable baseline is the only way to accurately measure how much control a patient actually has over their cognitive decline. Among all these non-modifiable risk factors, there is one that acts as the strongest overall predictor of Alzheimer's. That variable is age, and it operates as an exponential rather than linear threat to the brain. This graph maps the raw epidemiological data of Alzheimer's prevalence against patient age across global populations. Notice how the prevalence curve remains entirely flat for decades. Then, at age 65, the slope steepens sharply. Age acts as a compounding weight that progressively crushes the brain's pathological tolerance. Returning to this prevalence curve, we can track the specific scale of this failure in regions like Western Europe or the United States. By the time the population reaches the over 85 bracket, prevalence jumps dramatically, approaching 50%. In the terminal over 90 bracket, incidence rates, the volume of new cases, reach nearly 40% per year. Moving past age, we encounter the second major demographic vector, sex. Epidemiological data from both Europe and Asia shows a consistent, measurable bias toward female susceptibility to the disease. This higher prevalence is a distinct biological vulnerability, operating entirely separate from the statistical artifact of women simply living longer than men. These demographic vectors signal a systemic failure of A-beta clearance mechanisms over time. We can trace this failure directly to genetics, which splits Alzheimer's into two distinct phenotypes. The first is early-onset familial Alzheimer's disease. This phenotype accounts for only 1 to 5% of total cases, but it operates with absolute determinism. The disease here is driven by autosomal dominant mutations in 3 specific genes: amyloid precursor protein on chromosome 21, presenilin 1 on chromosome 14, and presenilin 2 on chromosome 1. Presenilin 1 and 2 act as the catalytic components of the gamma secretase complex, an enzyme in the cell membrane. When it cleaves the APP substrate, mutations in these genes force the overproduction of the toxic amyloid beta 42 isoform. This exact mechanism also explains why patients with Down syndrome, or trisomy 21, almost universally develop Alzheimer's neuropathology. It is a direct result of gene dosage. Having a third copy of chromosome 21 guarantees the overproduction of the amyloid precursor protein, forcing the rapid accumulation of amyloid plaques. For this patient population, Alzheimer's is an inescapable mechanical guarantee written into their genetic code. The vast majority of Alzheimer's cases belong to the second phenotype, late-onset sporadic Alzheimer's disease. Here, the genetics are probabilistic rather than deterministic. This genome-wide association plot visualizes the landscape of over 30 identified genetic polymorphisms linked to sporadic Alzheimer's. Among the scattered data points, there is a massive, undeniable statistical spike located on chromosome 19. That peak represents the gene for apolipoprotein E, or ApoE. Specifically, the ApoE epsilon 4 allele acts as the single strongest genetic risk modifier for late-onset Alzheimer's, and it operates on a strict dose-dependent curve. Possessing one epsilon 4 allele yields a 3-fold increase in the risk of developing the disease. Homozygosity, inheriting 2 copies of the allele, yields an 8 to 11-fold increase in risk and drastically lowers the expected age of onset. Secondary loci on the genome, like TREM2, point to similar vulnerabilities, specifically indicating a failure of microglial cells to clear debris. Sporadic Alzheimer's is the result of a probabilistic stacked deck. This genetic profile severely cripples the brain's ability to clear toxic proteins as it ages. How do we reconcile absolute genetic determinism with probabilistic risk? To build a model of vulnerability, we overlay these datasets. Early onset mutations bypass the age curve entirely, forcing pathology decades early. Conversely, late onset genes rely on the age curve, using time to slowly exhaust the brain's clearance defenses. These factors converge at the age genotype threshold. This is the specific biological breaking point where heavy, accumulated pathology finally triggers clinical dementia. The threshold gives way under immense, accumulating physical pressure. This immutable bedrock of age and genetics dictates the biological threshold every patient crosses, establishing the exact narrow window we have left to intervene with modifiable lifestyle changes.